Now, this is my last presentation for you. We bracketed my 4 presentations. Super complicated. The stuff we did Thursday night, the biblical worldview, and the fact that we're actually made of empty space. And then we went, what Darwin didn't know, history. And then we did the last one, races, a little bit complicated, a little bit easy. Hold on to your socks. I'm going to pull out all the stops here. I'm not going to apologize. What we're going to do is take a look at the complexity of the cell. And I'm sorry God created this, we don't understand it. The point here is not understanding, the point is shock and awe. When we consider what God has made, we're not going to understand it. We're never going to know everything that God knows, and we're never going to be able to recreate what God did. We're not that smart. All right? All right. I'm going to take the human genome and compare it to a computer. Man's best computers. And man's best computers are not even remotely going to compare. That's how much complexity we're talking about. But this is going to be fun. Now, as I said earlier, my background is in marine biology, and genetics, genetic engineering. I am a practicing geneticist. I love studying DNA. It's my passion. I was writing computer programs on the plane on the way over here. I've actually been sitting there now for 3 days, burning a hole in the back of my mind. I can't wait to get back and do mad science, pounding on keyboards and cackling over numbers. But we're going to be talking about the DNA molecule itself, which, as far as I'm concerned, is the most complex information storage mechanism in the known universe. It is a long and fragile string. You have this much DNA inside each one of your microscopic cells. That already tells us God is an amazing packaging engineer, because I could not do that. If you were to take your DNA and make it as thick as a hair, you'd have 32 miles of DNA packed into something about the size of a golf ball. And on that DNA is a code that can be read and used. So if I had a microscope and someone had written letters or a message on these hair-wide DNA strands, I could open up the golf ball and look at it through my microscope and finally find the message, right, and do something about, bake chocolate cookies or whatever the message is on there. But I would make a disaster. I would make a bird's nest. I would ruin it. I would not be able to do it twice, because 32 miles of hair would be really, really, really hard to keep from getting into a tangled mess. That's you. And you have about 60 trillion cells in your body with that much DNA. Now the genome, it's a big word, that's just a sum of all your genes. It's broken down into discrete pieces, we call them chromosomes. You inherited 2 sets of chromosomes, one from your mother and one from your father. So there's a backup copy. That's really smart. When they used to launch a space shuttle, every critical component that they could duplicate, they did. In fact, they triplicated it. There was 3 main computers for each type of computer. So they had the main one, and if it broke, they'd have a backup copy, and then they had a backup to backup. That's called redundancy. Redundancy is an engineering principle that's very important. If you can duplicate something, you do. Now, when they build bridges, they don't duplicate the bridges. They just make the bridges a lot bigger than anyone would ever think they would need. They're over-designed to be robust. And we see duplication and over-design in the genome like any good engineer would apply. It's not something predicted by evolutionary theory, but it is predicted by common sense. Now, this image is actually a photograph of a person's genome. Because when the cells are divided, the chromosomes condense. And you can take a picture of the condensed chromosomes and then cut out the pictures and line them up with a karyotype. This is the way they used to diagnose Down syndrome. One of the pieces of one chromosome would be duplicated and copied to another. One of the chromosomes would just be a little bit too long. Hey, that's weird. That's not right. And all the people with Down syndrome have that. This was painstaking work. This is very, very difficult stuff. Today, though, we have gene sequencing. We don't have to do this. We just give you a picture of a genome. Now, interestingly, that person was a man, because there's a Y chromosome there. All the chromosomes were paired except for the last two. They're called the sex chromosomes. If you have two X chromosomes, you're a woman. If you have a Y chromosome and an X chromosome, you're a man. I remember in graduate school I was taught the Y chromosome is a vestigial piece of junk DNA. It has almost no genes on it. If you lost a gene, the Y chromosome, it wouldn't even matter. Men are superfluous. Anyway, the point is, they thought that the Y chromosome did almost nothing because it has almost no genes on it. In fact, a lot of times when it's condensed, it's actually bent. It looks broken. It's just junk. Well, now since we sequenced the human genome, we know the Y chromosome is a master control switch that affects genetic expression on thousands of genes on all of the other chromosomes. The presence of the Y chromosome has a profound effect. Men and women don't look the same, don't act the same, and don't think the same. We're very similar, there's lots of overlap, but on average there is a difference. And the outward genitalia of male and females is profoundly different. In fact, there's no stage of development. If there's a Y chromosome present, that embryo is going to be a male. There's nothing to stop it. Early in development, though, there's no differentiation between male and female. The embryo is developing, and you can't tell the difference. It's not like, oh, the embryo is female at first. That's not true. It's nothing at first. It's in between. Now, there's a Y chromosome. At one point, the Y chromosome turns on and starts dosing the embryo with bursts of testosterone. And that reprograms the cells to do different things. Interestingly, there's one part in development. Early on, your reproductive cells of the future generation are not in the embryo. They're outside the embryo. They're sitting on the allantois, outside of the little baby. The baby's growing and growing. They're just sitting out there, not dividing, because the more you divide, the more mutations you get. These cells don't divide. And then at one point, they form a little Congo line. They march across the outside of the embryo. They invade the embryo, find the gonads, and when they invade the gonads, that's when you have an ovary or a testes. That's so cool. Development and genetics and embryology is amazing. But genetics. We've now sequenced the human genome. It cost the US government about $3 billion, took about 10 years or more. That's about $1 per letter. Today, there are thousands or tens of thousands of fully sequenced human genomes. The $1,000 genome is not just here. I had my genome sequenced. It was a Black Friday special, Dante Labs. I think it was $325. $3 billion in the year 2003. $320. And they said 2 years ago I did it. So for 20 years, the price dropped by 1 billion-fold. That's how fast the technology is progressing. And we have so much data today, we're awash in data. This is like one of the first times in human history we've had more data than we have theory. And it's really exciting to be a geneticist. And to be a geneticist, you have to be a computer programmer. You have to deal with Amazon Web Services and distributed data because you can't fit the data on a personal computer. There's too much data. And we're all crunching numbers like crazy. Which means it's a field day, and there's a lot of low-hanging fruit out there. And some enterprising mind says, hey, I think I can explore something. It might not have been explored yet. And a lot of new discoveries are happening. It's really, really, really exciting. I'm going to tell you, the human genome is not simple at all. If it were simple, evolution might be possible. It's not simple. In fact, the complexity of the genome is the opposite of what evolutionary theory would predict, because random processes don't produce complicated things. It might, maybe, produce something simple. I'm also going to claim that the genome is also not static. It operates in 4 dimensions. Okay, remember math class? What's a one-dimensional object? A line. A 2-dimensional object, a piece of paper, length and width only. A 3-dimensional object, length, width, and height. Right? What's 4 dimensions? It's something that changes over time. The 4th dimension. Now, this is a 3-dimensional object. It doesn't change. A movie is a two-dimensional screen projection that changes over time. That's a third dimension. You and I are experiencing each other in four dimensions. There's length, there's width, there's height, and there's time. The genome operates in four dimensions. I'm going to explain each dimension at a time. Now, comparing this genome to a computer produces very interesting results. Here we're comparing the simple, the simple bacterium, Escherichia coli or E. coli, to the Linux operating system. On the left, on the top, those are all the genes that are found in the bacterium. In the middle are a set of genes that control the other genes. And on the bottom are all the proteins that the bacteria produces. Interesting, huh? On the right, we have computer programs and all their brilliance make a lot of high-level programs that control a myriad of subroutines that output a minority of output on the screen. One of those might be controlling a disk drive, one might be changing a pixel, one might be listening for keystrokes, or all things computers do. But we have this massive architecture, all this programming to do just a few things. Bacteria have a few things that can do a lot of things. So already we realize that our best technology is upside down. Life is not just designed, it's designed very well. And that's really cool. We do things inefficiently because we're not as smart as God. So the first dimension of the genome is a linear string of letters. If you were to look at like the Y chromosome, it reads CTAA, oh boy, that's so boring. It really is boring. In fact, that goes on for 60 million letters. You heard a lot of repetition in those letters I just cited to you. There's a lot of repeating things in the genome. That's part of what they call junk DNA. But there are millions of stretches, of long stretches of junk DNA. Except, is it really junk? What if it's three-dimensional scaffolding? What if it's designed to curl into a loop that holds two genes in a specific place in space? You can't delete all the junk DNA in the human cell. The cell won't work. There's a structural component. So just because the DNA doesn't code for protein doesn't mean it has no function. Do you know what a vestigial organ is? Charles Darwin and his contemporaries, they had made a list of like 80 vestigial organs in the human body. These are evolutionary leftovers. Things that don't do anything in our body, like the thyroid, the thymus, and the appendix. Ridiculous. We know so many functions for the appendix, we don't know what its primary function is. Tonsils. I had my tonsils opened up a couple years ago. I had a really bad problem with sore throats. And I said, all right, doctor, I guess you got to do an appendectomy. He goes, no, we don't do that anymore. What? Because we're just going to cut holes in it and help them drain better. Why? Because that's part of your immune system. One of the primary beginning parts of your immune system for your entire alimentary tract is your tonsils. They don't want to take them out. They're not vestigial. Junk DNA is also not vestigial. But DNA is really boring. You cannot just read the human genome. No one's ever done that. No one would ever want to do that. Why would you just sit there and read, billions of letters and they just mean nothing. So, some friends of mine, they wrote a computer program, which I love. Let's replace each of the letters with a colored dot. A, C, G, and T, 4 different colors. That's the same exact data I just showed you. Do you see repeats? Let's turn on my laser pointer here. This area in the middle is a gray area that until just like a month ago, we didn't have sequenced. The human genome was not completed until very recently. 20 years of incompleteness because there are repeats. And some of these repeats are perfect repeats. The same exact letter set again and again and again. And the machines that we use to sequence DNA, they only sequence about 300 letters at a time. And what you do is you sequence them all at random, and you take the pieces and you line them up. But if you have a repeat, you don't know where in the repeat to align them. It's like you get in the car, you turn on the radio, your favorite song is playing, la 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 la, and you mess up because it's in the middle of the song, not in the beginning, and you started singing in a repeat. Right? Our music has lots of repeats in it, and during the refrain, it's the same exact refrain, but the verses are different. So if you're in the repeat, you don't know where you are. Same thing here. If you're in a repeat, you can't do that. So we needed long distance. You had to sequence millions of letters, not 300 letters, before they could actually paper over these gaps. But looking here, there's a repeat here, and you can see it hiccups every once in a while. It skips backwards a little bit. There's little deletions in there. There's a repeat here. It's this long, 1, 2, 3, it's like 4 repeats in one string, and then another. There's another repeat at different length. That's really cool. That's the first dimension. Is everyone happy with that? The second dimension is the way DNA interacts with itself. So if you had a piece of paper, you could write out all the letters of the genome and say, oh, well, this gene interacts with this gene, draw an arrow. This gene interacts with that gene, and draw an arrow. And you need a two-dimensional piece of paper to represent all that. One of the classic two-dimensional things that we study in biology is called a lac operon. Lac is lactose. It's a sugar. E. coli can break down lactose, but in the presence of glucose, they don't want to break down lactose. So they can turn the system off, or they can turn it on. There's 3 genes, LAC-Z, LAC-Y, and LAC-A, and they're right next to each other. Upstream of the genes, things can land on the DNA that can interfere with the production of those genes or turn on the production of the genes. Those things are made somewhere else in the genome. It's a two-dimensional thing. You draw out the whole genome of E. coli. These genes over here turn this gene off. This gene over here turns the genes on. The two-dimensional system. This led to a lot of mistakes in biology. One of the things is called the one gene, one enzyme hypothesis. Because in bacteria, this gene makes one protein. That protein does one thing. You only have 22,000 genes, but you make about 300,000 proteins. There's not one gene, one enzyme. Using bacteria as a model for humans was a big mistake. It actually slowed science down for a long time before we finally realized, wait a minute, we're more complicated than bacteria. But if you were to take all the DNA and connect all the places that interact, you wouldn't be able to see the DNA. There'd just be nothing but red arrows on the screen. Okay? Two-dimensional networks. There's also two-dimensional stuff that happens in these so-called junk DNA. This is called a microRNA regulatory network. MicroRNAs are little pieces of your genome that are made into RNA, but they're not made into protein. This is a network of genes that affects the hardening of the arteries, or atherosclerosis. The genes are there in red. And all of these little things sticking off, these are little marker RNAs that interact with the genes. These things are in the so-called junk DNA. It's not looking very junky to me. Just because it doesn't make protein doesn't make it worthless. In fact, you are not a DNA computer. You're an RNA computer. Your cells make massive amounts of RNA. Some of that is turned into protein. The RNA is the thing that controls which proteins are made and when and how much. It's like if you were a computer, the read-only memory on the chip, that's your genome. The random access memory, that's the RNA. The output is the proteins. In this case, I'm looking at a screen. The output for this computer is a screen. Your output is proteins and sugars and things like that. But most of the work in your genome is not done on the level of DNA. It's done at a higher dimension. All these decisions your cell's making is happening in a level that we can't sequence because the RNA is made from DNA and it's usually modified by a whole bunch of different things and changed around, split and divided and now it can control whether or not you're going to be diabetic or not. There's this level of abstraction between the human being and the genes, and it's huge. When they decided to sequence the human genome, the scientists convinced the government to spend $3 billion by promising, we will cure a disease. And they cured no diseases. because we're much more complicated than just DNA. So the second dimension, there's specificity factors, enhancers, repressors, activators, transcription factors, on and on and on. It's like, it's so complicated. That's the point, it's complicated. Then something else happened. Right after the genome was decoded, the ENCODE project started producing results. What they did was they said, we want to see how much of the genome is active. Let's look at 1% of the genome, which is 30 million letters. That's a lot of letters. That's almost the size of the Y chromosome itself. Let's just look at 1% of the genome, and we'll take pieces all over the genome, and let's see how many times this piece of DNA is turned into RNA. Whether it's turned into protein or not, we don't care yet. Let's just see how much. They found out almost all of it was turned into RNA. What they found was that the average letter in DNA is turned into 6 different RNAs. Let me give you a little video clip from my Evolution's Achilles Heels movie. There's a piece of DNA, there's a ribosome zipping back and forth, making copies of RNA from one strand or the other strand in opposite directions. And notice they don't all line up the same. They're different stop and start points. That one, in this example, that one letter of DNA is included in 6 different RNA messages. One of these might affect how well you see in the dark. One of these might affect if you hiccup after you eat garlic. They have nothing to do with one another. But if you change that one letter, you potentially affect multiple different features. That's really tricky, because natural selection can't deal with that. Natural selection is a clear target, a very clear target that's strong enough to influence how many children some offspring an organism is going to have. It needs to be a powerful effect for natural selection to see it. And if it's affecting more than one thing, I mean, what's it going to kill you for? The fact that you don't see well in the dark, or that you can't digest garlic? What if both of them are important? And all sorts of other random things. The point is this. When God wrote the genome, He did something no one predicted. He took information and He interleaved it. He wrote strings of information on top of each other. So each section of the genome does more than one thing at the same time. I can't write computer programs like that. There's no way. If I start at position number 50 in my computer program, I have a chocolate chip cookie recipe. But if I start at position number 51, I have a recipe for rocket fuel. No, we don't think that way. Biology does. Natural selection does not explain that. If you look at a typical gene, DNA is double-stranded, so we have 2 strands of DNA in blue, but genes are only on one strand. And we tend to have, I color it yellow here, an untranslated region upstream of the gene. That's where all the controlling things happen. The enhancers, the repressors, the activators, they stick on the DNA upstream of the gene, and that tells you whether the gene can be turned on or turned off. At the other end, there's another untranslated region and other features that influence the gene, but also tells the ribosome when to let go. Gene's over now, you can let go, and so it'll go find another gene to translate or transcribe. But in human genes, in higher organisms, not in bacteria, all the more complicated organisms, our genes are broken up into pieces. We have introns and exons. An exon is a piece of DNA you can cut out and throw away. Of course, sometimes they contain genes themselves, and sometimes they have other regulatory features, but for now, you cut it out and throw it away, and then you take your exons, or introns are thrown away. The exons are joined together to make a gene. And then you can translate that and make a protein. Except, in between the introns and exons, and the ends of each one, are a myriad of little short 10 or 13 letter codes, dozens of them per joint, that say, use this exon in this gene at this time. But don't use it in this gene at another, use it in a different gene at a different time. This is how we can produce hundreds of thousands of proteins from only tens of thousands of genes. They're spliced and they're diced. There's not one gene, one enzyme. In fact, there's no definition of the word gene anymore. In bacteria, maybe, but not in humans, not in mice. Our genes are extremely complicated. So it might be like the eyeball manufactures this specific protein, but the liver manufactures a different version because it has a different exon. But only after you eat garlic. The other times you use a different exon because you just ate, I don't know, asparagus. Your proteins are manufactured differently over time, the fourth dimension. In fact, you don't need the same proteins as an adult as you do when you're an embryo developing in the womb. And different proteins turn on at puberty, and different proteins turn on at old age. The third dimension of the genome is the three-dimensional shape. The chromosomes, typical shape of a chromosome, you have a string. In the middle is called a centromere, a bunch of repetitive DNA. On the ends you have what are called telomeres, also repetitive DNA with a different repeat sequence. And every time a cell divides, the telomeres tend to get shorter. When they get totally short, they don't divide anymore. So death is pre-programmed. But in the testes, telomerase is very active, and in the testes, telomeres get longer. That's why each generation doesn't go extinct, because the telomeres get lengthened again. If you just look at the genome, you might think that you just shove all the chromosomes into the nucleus, right? Let them do their thing. But it's not the way they are. They're not randomly distributed. In fact, all of the centromeres are clustered in a place called the nucleolus. The telomeres are anchored to the nuclear membrane. So it's not junk, it's structure. In fact, if you take the DNA from the top left there, the double helix, it's wrapped around histone molecules, which are proteins. And there, you ever take one of those balsa wood paper planes with a propeller and a rubber band? And you turn it, you turn it, you turn it, you fly it, it's really cool. And you, oh, we gotta go really far. You turn it, turn it, turn it, turn it, turn it, and the rubber band snaps. Well, you know how after you get it all the way twisted up and you keep turning it, it kinks, makes a twist of a twist? That's what DNA does. You have twists of twists of twists of twists, and it condenses down into a chromosome that you can see. Usually, though, during most of the cellular lifetime, the DNA is diffuse, not condensed. And you might think it's just thrown into the nucleus, but it's not. In fact, I love fluorescent DNA tags, love it, part of my doctoral research. If you take the chromosomes and stain them each a different color, you can look inside the nucleus and see where the chromosomes are. They each have a predefined position in 3D space. One of the first things that geneticists did after the genome was sequenced, they said, hey, let's look at genes that are used together. Because remember the lac operon, those three genes are right next to each other? So they made a hypothesis. We predict that genes that are used together will be found next to each other in the human genome. They're not. They're willy-nilly distributed, different chromosomes, different places, different directions, and they said, it's just a bunch of junk. Millions of years of evolutionary experiments led to all the genes being randomly associated with themselves in the nucleus. Then someone decided to stain them all separately and fold it up in 3D space, and they realized genes that are used together are found next to each other in 3D. Even if they're found on different chromosomes, when the chromosomes are together, genes that are grouped together are right next to each other in 3D space. So whoever programmed that linear string of DNA knew how it would fold and how it would be organized in the nucleus, or else the system wouldn't work. Because when you turn on a biochemical pathway, you need all the genes to turn on, so they can all turn on at the same time. They tend to also form a little pocket in the nucleus, and it's right next to a nuclear pore. So all that messenger RNA can be at the same time, sent out the same pore. And something else, we knew that there are different classes of ribosomes, and some ribosomes work good with some piece of DNA and better with other, like if there's more C's or G's or more A's and C's, or A's and T's, the different ribosomes will be found there. Well, now we realize that clusters of genes that are used together in biochemical pathways tend to have the same patterns. And the ribosomes are tuned to work with some types of genes and not others. So now we're talking about a finesse in the genome. We're talking about fine-tuning and exquisite precision engineering. There's no junk here. There's no mistakes here. This is nothing but brilliant engineering principles applied to a very complicated system. And humans didn't think of this because we're not smart as that. We discovered it, but we're just discovering what God did. And it's all contradicting evolutionary history. We just found that out. All right. The fourth dimension is how things change over time. So when you need a gene to be expressed that's not being expressed at present, it's probably buried in your nucleus. Well, if you want that gene to turn on, you've got to change the shape of the chromosomes to expose that gene so you can make copies of it. And when you're done, the chromosomes fall back again. Cool. So there's changes in the shape of the chromosome over time, which is the fourth dimension. But not only that, so the 3D architecture changes. The interaction patterns, the two-dimensional thing, that also changes. Because all of a sudden, hey, I got to turn this gene, so this thing over here says, okay, it makes something new, and it comes over here, and the interaction patterns change. But not only that, the linear string of letters can also change. And this is mind-boggling. Computer programmers gave up in the 70s of using dynamic programming, where they would have the computer program reprogram itself. No, stop, don't do that. Have each thing independent so we can control it and predict what happens. When you start having a computer program reprogram itself, it just uses you and destroys itself. But the genome reprograms itself. When you're developing in your mother, your brain is making millions of neurons a minute. If I was going to develop a brain, I would lay down a sheet of brain tissue and then make it thicker. That's not how God designed the brain to develop. Your brain cells develop in specific brain cell forming regions, and the brain cells migrate through the developing brain to get to where they're supposed to go. And brain cells have different genomes. There are things called retrotransposons, or jumping genes. They're a little piece of DNA that can have specific things in their end. If they touch, they can form a circle and pop out, and go somewhere else and pop in. They can move in the genome. The DNA reprograms itself. And very often those loops of DNA have the programming to turn a gene on or turn a gene off. And as they migrate and through the genome, they're literally changing the genome. Your brain cells have different genomes. Your liver. When your liver is being made, one of the ways you control gene expression is by copy number. So if you want a lot of something, you have a lot of those genes. If you want a little, you only have a couple of those genes. Well, the liver needs a lot of copies of detoxifying and breakdown genes, enzymes that do work. But your eyeball doesn't need that many copies. So the genome in the liver makes extra copies of the chromosomes. You don't have the same genome in all your cells. There's a retrotransposon that humans have that controls the binding of the placenta to the uterus. So-called piece of junk DNA. If you didn't have it, you'd have no babies. There'd be no mammals. There's another piece of junk DNA retrotransposon. I think humans have it too. But in the mouse, if you turn it off, you delete it out of the mouse embryo will develop and then stop. Basically, a piece of so-called junk DNA is required for the development of the mouse. It's not junk DNA. It's fully functional and very important components of life. So the fourth dimension, we're talking about conformational changes during development, over minutes when different cells need to detoxify things and do new things. We're talking about rearranging, duplications, and deletions, and we actually don't know how dynamic the genome is. It's not static. So this is a computer program. I predict that computers in the future are going to change. Because as we learn about how the computer inside our cells works, that's a great idea. I think computer architecture is going to change also when we realize what we can do in four-dimensional space. Just one more interesting little tidbit. Genes turn on and genes turn off over time, depending upon your environment. This is a study done with men with enlarged prostates. Now, they didn't have prostate cancer yet, so the people could do this experiment. What they did is they took a biopsy, and they told the guys come back in a couple of months, and they took another biopsy. That's the left and the right on this chart. And for this group of men, they had a control group, they said just come back in 6 months. For this group of men, they just gave them general diet and exercise routines. Nothing extreme, no South Beach diet, no go run a marathon. Just dudes, eat well and get moderate amount of exercises. And in this chart, on the right is before, on the left is after. Green is off, red is on. The top are, that's opposite. See this one? On the top are stress genes. Genes that are expressed and turned on when you're under stress. Beneath the line, the majority of these genes, these are just general housekeeping genes, happy genes, genes that just work when everything's working normally. What we see is a dramatic shift between green and the red. These stress genes turned off. The happy housekeeping genes turned on. Or their expression went up, their expression went down. We don't need any more information than that. We know what controls aging. We know what controls lifespan. We know what controls health into later lifetimes. It's general diet and exercise. It's not running marathons, it's getting off your rear end. Okay, say my mother. She has no cartilage in her knees. She's totally losing all of her muscle mass. She doesn't move. And it kills me. She's 83. I know other 83-year-olds are still spry and moving around, but the body does age. We know that. The body also will turn itself off. There are genetic factors that control lifespan. We know what they are. We control a single letter in a mouse, we get double its lifespan. You can control how much the mouse eats and what it eats, and you can double its lifespan. So could people in the past have lived for hundreds of years? It depends on their genetics, it depends on their lifestyle. Yeah. But we now can see today, to improve the quality of life, eat well and get off your rear end and move. And that will not necessarily add years to your life, but improve the end. My goal in life is to do a swan dive at the end. That's my goal. Go out in a blaze of glory. Now, that might not be what God has in store for me. I understand that. I'm starting to develop arthritis in my spine. I'm constantly doing this. If you ever notice me sitting, my back hurts. I can't quite sit up straight anymore. Oh no, I don't want to go out with that. But it might happen. Just saying. We can do a lot of things to improve the quality of our life over time. And the reason I'm saying this, I don't care how long I live. I just don't want to squander my remaining years when I can be used by God effectively. If I'm a couch potato, I'm not out talking to people. And if my mind starts to fade because I'm not working with memory exercise and things like that, I might be able to, even email my kids and things like that. So let's just take the bodies and the brains that we were given and use them as best we can. We have enough information. We know. Don't drink Coca-Cola. Sorry, it's poison. Why? I love Coca-Cola. I don't know, once a year, might have a little, maybe once every 10 years now. I don't know. There's no reason for that kind of stuff. Of course, I did have some of those, I can't pronounce them, the TJ something in them, the little fried things with the butter and the strawberries and cream. So I'm exercising today by walking around, so at the end of the day, I'm going to get some more. We see all of these changes over time. Fourth dimension completely gets rid of the junk DNA hypothesis. Forget it, it's not true, it's dead. Now, let me read a quote from a geneticist. The failure to recognize the full implication of this, particularly the possibility that the intervening non-coding sequences, that's big speak for junk DNA, may be transmitting parallel information in the form of RNA molecules, may well go down as one of the biggest mistakes in the history of molecular biology. Junk DNA is dead. The evolutionist does not want to admit this. They need it, because without it, the genome is too complicated and they can't explain it with evolution. But it is dead. So, what is the genome? It is a multi-dimensional operating system for an ultra-complex biological computer. Okay, so walk up the stage right now. That's all you need to know. But it has built-in error correction and self-modification codes, and multiple overlapping DNA codes and RNA codes and structural codes, or DNA genes or RNA genes. It was also designed with a large amount of redundancy on purpose by a highly intelligent being. He used sound engineering principles during his construction. But despite the redundancy, it displays an amazing amount of compactness, because only about 20,000 or so protein-protein genes make over 300,000 distinct proteins. So my challenge to the evolutionist is simply explain the origin of the genome. I'm going to read a quote from Darwin. I know this is out of context, and I know the evolutionists jump on us every time we try to say this. But he said, if it could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. He then goes on just to try to explain how things can happen, but this is, his quote. Well, my claim is that the genome could not have arisen through no naturalistic processes. So, Mr. Evolutionist, I want you to give me a workable scenario, including the source of the informational changes, an account of the amount of mutation necessary, and how much natural selection is necessary. I'll even give you millions of years. I don't think it can be done. The mathematics and the complexity of the genome tell us otherwise. So just something for you to have in the back of your mind as you're talking to people. Understand, God is amazing. He built us in amazing ways. The person you're talking to is amazing. They're not simple and they're not junk. A lot more. Look at Creation Magazine if you like. We've got 45 years now, Creation Magazine has been published. Started in Australia many years ago. I don't even remember. I mean, we have tens of thousands of people subscribing to this around the world. It's very important, very powerful information. It's there for you. If you'd like to get it, if you sign up for a 2-year subscription, we're also going to sign you up for our digital magazine, which will send an email out with a link, and you can send that to anybody who wants. So they can read your Creation Magazine on their laptop, their phone even, or their tablet. But for a 2-year subscription, we'll also give you a copy of Darwin, the Voyage That Shook the World. It's our first movie we ever made in honor of Charles Darwin's 200th birthday. That's my talk the other day. We're also going to throw in a copy of Fallout. It's about a 35-minute mini documentary we made. We took a TV camera to college campuses, multiple college campuses, with about a 2-hour drive of Atlanta, and we interviewed hundreds of students. And we asked them these questions. Were you raised in church? If they said no, we said, see ya. Oh, you went to church growing up? Were you ever taught anything about creation and evolution? The next question was, you still go to church? And what we found was a very strong formula. The churches that were preaching this from the pulpit, the Sunday schools that were discussing this, families that discussed it over the dining room table, they were raising students that were much more firm in their faith and able to stand tall in a very anti-Christian, hostile environment called the college campus. The families and the churches that didn't deal with these issues, with one exception, every student that said, I never talk about anything about creation and evolution, none of them were still faithfully attending church. So it's not perfect, it's statistics, right? We all know the statistics, but there's a very clear trend. Engage the big issues, you raise mature Christians. Most people we're going to talk to know nothing about creation and evolution. Those that do kind of have, most of them have a skewed version of creation and evolution. Oh God, that's just the way God did it. Things like that. Very few get a good, seasoned, reasonable defense. If you like Creation Magazine, the sign-up form's going to be at the table there. It's really simple to get it. Just click, done, and I'll take care of it. You also might want to look at Mitochondrial Eve and the 3 Daughters of Noah. This is my first genetics talk that I gave to creationists. I did this in front of 700 people in Australia. I call it Mitochondrial Eve and the 3 Daughters of Noah. I didn't think of a good title. I should have called it Genetics of God, or God's Genetics. Basically, I'm saying that geneticists have discovered evidence of Adam and Eve, Noah's flood, and the Tower of Babel is in the human genome. And I just took what the evolutionists discovered and presented it on stage. It was fun. This talk, the high-tech cell in a very more polished format, I didn't stumble nearly as much, is also available. In fact, this is my favorite talk I've ever given, ever. And it just happened to be the first time I gave it, and we happened to have a TV camera present, and so we turned it into a DVD product, and it's out there on the table if you're interested. Here's Evolution's Achilles Heels, the book and documentary. Powerful material. This is 3 years of my life that I poured into this. This was us making the evolutionists defend from their gold, right? So our best arguments against evolution from PhD scientists. We won 2 different awards for our documentary. In fact, we presented at the San Antonio Independent Christian Film Festival. There were 13 documentaries, and we lost to Mike Huckabee. Just close. But we got second place. I'm very, very happy about that. That's out there if you're interested. The book is also excellent. Genetic Entropy and Mystery of the Genome also deals with the new cover. Also deals with evolutionary theory, what the evolutionists believe, what they actually can say on the side, by Dr. John Sanford, a good friend of mine and a world-famous scientist. And again, I'm going to leave you with this. 1 Peter 3:15. In your hearts honor Christ the Lord as holy, always being prepared to make a defense to anyone who asks you, for reasons of hope that's in you. You do it with gentleness and respect. We can do that. When we're engaging people, we just have to understand how complicated they are, how wonderfully created they are, how beautiful they are, and how they reflect God's handiwork, even though they don't understand that themselves yet. You got it? Let's pray. Father, thank you so much for the genome of all things. Thank you so much that we get to sit back and say, I don't understand. But that's okay, because we're looking at what you built, and we can just marvel and awe at your brilliance and your creative genius. Thank you for that example for us. It's encouraging, it's exciting. No, we don't get it, because we're not you, and that's okay. Help us, Father, to continually fill our minds with awe of what you've done. To continually put ourselves in the right perspective. As created by you, as loved by you, as engineered marvels of creation, but also as emissaries of you and your kingdom here on this earth. The people we interact with are equally wonderful and beautiful and creative, but sometimes it's hard to see that because we see the aggression, the anger, and the disdain for what we're trying to do. Help us see past it and love them, because they are also your children.